in terms of reading the paper, there are certain things we want you to get out of each of these papers in order to make an annotation. And what we've actually done is we've provided you with a template, which is this little table that's in your Google Doc. And the idea is every time you read a paper, you're going to copy this table and fill it out for each one. And so the idea is that, first of all, you're going to list the, what the paper is, so we know what it is. And there's something called a PubMed ID, which most of you guys will know about, and we'll talk about later. And it's just a number, it's just a number that allows us to know exactly what paper it is in the PubMed database. So that will be there, and then we want you to then get a sense of what the big picture questions are for the paper, and then what the specific questions are, and then as you go through each figure, get a sense of what it is they're trying to do, what techniques were used, what the conclusions are, and then we're going to try and think of that in the context of what does it actually say about function. Because there's going to be figures in the paper, in the papers, even if the paper itself says something about the normal function of a gene, some of the figures will be relevant to what we're trying to do, and other figures are sort of background information that may be interesting in science, but it has nothing to do with determining what the function is. And we'll see some examples of that as we go through the paper. Okay. All right, and then there's going to be a link here to another article, somebody else's perspective about how to do the paper. All right, so each of you guys should have a Google Doc that has, a copy of this, and then you can copy and paste the whole table in different ways. And people have found that this really helps them organize the reading. Okay, so the next thing is we're going to actually go over a specific old paper. This is just an example of a paper. It's about a gene function. And we're going to both show you how I read the paper to extract information from it. And then also just illustrate this thing called Google CAMI. So has anybody used Google CAMI before? Okay. So you guys probably have all read PDFs before, right? And the idea behind Google CAMI is that the whole class, or whoever you want to share with, can share a PDF. And you can put notes and highlights on it, and other people can see your notes and highlights, and you can see their notes and highlights, and you can sort of use it as a way of, it's part of the idea of working with teams, is you can actually share stuff and say, what do you think about this, and so on. Right. So if you go to that Google CAMI link, you'll see this is a paper that we've done before. We haven't figured out how to unmark all the people who looked at it last year. So I should be able to do click on this, right?
Mm-hmm.
I guess it looks like this. Okay, so this is the Google CAMI view. And I can zoom in on it. And this is the paper, which is about topoisomerase 4. But first I need to give you a little bit of background. All right. So this is a paper about characterizing the genes that code a protein called topoisomerase 4. And so before I can tell you about it, first I have to tell you what is a topoisomerase. And you might have heard about this in some biochem or molecular biology class. So the idea is that a topoisomerase affects DNA topology. So you guys all know that DNA is double-stranded, antiparallel, like this stuff, in forms of double helix. And if you have a DNA that is circular, it can have what are called supercoils. Has anybody not heard of supercoils before? It's all heard about supercoils. Okay. So if you have a circle that doesn't have any supercoils, it just has the same number of turns in it as the double helical turns, that's called a relaxed circle. Okay. As you overwind it, then it can either have a positive or negative supercoils. All right. And so this is just a picture from a Wikipedia page or paper. You have these topoisomers that have different amounts of twist and ride, and then these different kinds of joints. Yeah, it's image modified from Wikipedia. So basically, the key thing is that topoisomerases affect DNA supercoiling, and they're actually targets for both antibiotics and cancer drugs because of the way they work, which we'll mention in a second. All right, so among the topoisomerases, there are two kinds. They're called, in a very cryptic way, they're called type 1 and type 2, right? Okay, type 1 topoisomerases cut one of the two strands and they swivel around the other one. So you can see how if you have an overwound circle, you can just swivel around this hinge and it will relax, and then it reseals then it will reseal the middle. And that's the type 1 topoisomerase. The type 2 topoisomerases actually cut both strands and they pass one double helix through the gap and then reseal it on the other side. So if you think about how this is top, you cut it and then it will wind up on the bottom. So that will affect the superhelical term. Okay? But it cuts two strands instead of one. Okay? And it turns out that what it does is this cutting and resealing, it's of course holding on to the ends, and it makes various covalent intermediates so that the ends don't drift away and get degraded and have horrible things happen. So the reason why, especially type 2 topoisomerases are targets for anti-cancer and antimicrobial drugs is that if you inhibit the right part of the reaction, which you can get if you get topoisomerase that will break the DNA and not be able to reseal it. Okay, and so in many cases, like if you have, say, a drug, many drugs affect cell wall abscesses, and for most of those, it's the idea is if you can't make the cell wall, then the cells will die. But you have to knock down the activity to quite a bit, because if there's a little bit left, you can still make the cell wall. Or there's different drugs that will affect translation by inhibiting ribosomes. But if you only inhibit half the ribosome, you still have stuff that can do translation and you can survive. The thing about topoisomerases is if you inhibit one molecule out of 100, that one molecule will break the DNA up into little chunks, which is very bad for genome, right? So it's kind of a dominant killing thing, which is why they're so effective as drugs. So the big question in this paper is, what is topo4 made of in E. coli? And then the specific question is, they had these two genes that were candidates for being the components of topo4. The genes are the products of the Par C and the Par E genes, and are they enough? Are they necessary and sufficient to make topo4? And then, based on the activity, what exactly kind of topoisomerase are we looking at? All right, so this is the actual paper. It's a nice sort of dry way. But it provides background on the different kinds of topoisomerases. And you see the domain is actually made marked to show the different places that matter. So the idea here is they had these two genes that they thought were the important genes, and their approach was to make the proteins, and being good biochemists, they were going to purify the proteins and mix them together and see if they actually have any topoisomerase activity. So if we look at various figures, this is a figure that shows that they can clone and sequence the genes. So that doesn't actually tell you anything about the function. It's just saying that we've got the right clone for the genes. So this figure is important for the paper, but it's not important for saying anything about the function. Okay. And similarly, this figure here is just showing they can purify the proteins. So they show that they can express them from some sort of plasmid or a cloning vector, and then they have biochemical purification, and they're just running a protein gel, which you guys probably get in some lab, right? Running the gels and blue staining, right, to see the proteins. And so they're just saying, okay, here's the purified one, here's the purified the other one. It doesn't say anything about the function. It just says, okay, now our starting material is good enough that we can actually do an assay for whether or not we have a biochemical function. So the first part that actually gets to is figure 3. Okay, and so and conclude that this is a functional assay is that this bold part which says relaxation and decativation activities of topo4. This word activities is a clue. Right? So you're looking for things that say activity. Well, in the figure legend, that's the sort of thing that can say, I've got a biochemical function that we can look at. And so what they're looking at here is based on the idea that they have two different assays, right? So one is that if you have these circles that have the supercoils, it affects the way that the DNA will migrate through an agarose gel if you set up the gel correctly. And so the idea is that if you have, or I believe that it's, okay, no enzymes, anyone. So if you start with supercoils, they will, because they're kind of knotted up, they're more compact, and so they go through the gel faster. And so you have this sort of thing where this is a supercoil and this is a relaxed circle. So you can almost never make supercoils without having some relaxed circles around too, because the DNA sits in the middle. And then if you have different topoisomers, if you have the right kind of gel, you can actually make this ladder for each of those different bands and a different number of supervalent turns. And what you can see is that here, as they're adding different amounts of the enzyme, which is a mixture of part C and part E, that you start out with just supercoils, and then as you get to enough enzyme, then suddenly it all goes up to relaxed circles after going through this intermediate step of making the different topoisomers. So that's basically saying that as we add this purified protein with the appropriate cofactors, which I'll get to later, that it will relax the DNA. That's one criteria for being a topoisomerase. Now, does that tell us if it's a type 1 topoisomerase or a type 2 topoisomerase? No, you can't tell, all right? So, in order to tell whether you have type 1 or type 2, you have to distinguish between making a double-strand break or a single-stranded nick. Okay, and that's where this assay comes in, which is called the decatenation assay. And this is a little bit obscure, because it relies on this piece of biology you probably don't know. There's this thing called a kinetoplast, which is kind of like a mitochondria from a weird parasite. And in the kinetoplast, I think Dr. Kuzreus wrote some of these. I think this is in
Trypanosomes?
Trypanosomes. So mitochondria have their own genome, and it's a little circle. And in the case of trypanosome mitochondria, which are called kinetoplasts, the circles, each mitochondria has like instead of having just one circle, it has a whole bunch of circles, and they form like a chain. So like the circles will be interlinked like this to make chain mail. Okay? And so they're not covalently linked, but they're topologically linked. They can't pull apart from each other, which is why, if you try to run them on the gel, they get stuck way up here at the top because they can't enter the gel because it's this big network of links. Okay? You can pull the circles apart if you take one circle and you make double strand break and you pass the strands through and you reseal it. Okay, that's called a decatenation assay. So this is catenated, this is decatenated. So famous magic tricks, right? Okay. So this is a decatenation assay where you start with catenated, and just like here, you're adding more and more of the enzyme, and now you get these black circles that come out, and that shows that. Now, if you just nick the circle, would you be able to pull them apart? He's shaking his head. How are you thinking? No. So the decatenation assay distinguishes between type 1 and type 2 topoisomerases. Okay. So from this figure, between these two things, this says it's a topoisomerase, this says it's a type 2 topoisomerase. So now we actually have a biochemical function for these proteins put together. These pure proteins mixed together with these substrates show that we have a type 2 topoisomerase activity just made of these two proteins. That's what biochemists do. Purify stuff, you put it back together and see if it does an activity. And this is the particular activity that they happen to measure, these topoisomerase too. Okay, does that make sense? Right? This looks more complicated than it really is, right? The next thing they did was they asked, does this require ATP? Okay. You guys have wondered about ATP, right? Entities need to do things. And surprise, surprise, it means ATP. There are some topoisomerases that don't require ATP. This one does. That means we have an ATP-dependent DNA topoisomerase activity.
So we filled out the rest of the paper table because if we were reading this paper, we would want to go through all the figures to find out what the authors have to say. about all of the different assays that they did. And so we've filled in the rest of this table so you can see an example of what it would look like. So just like we just walked through on figure 1, there were 2 panels and it showed nucleotide sequence and sequence prediction, and we concluded, okay, great, they could clone the genes. And for figure 2, they showed that they could purify the proteins. For figure 3, that's the one that we spent the most time on.
Right.
And
Is a type 2 activity, right? That's what we do.
Right, so we say what they did and what the conclusion was for each part of the figure.
Right. This is the ATP dependence, right? Then figure 5, which we didn't talk about, is essentially the same thing, but just they used other nucleotides besides ATP and showed that they didn't work. Okay. So it's not just any nucleotide.
So there are a whole bunch more figures actually in this paper, not relevant to the example that we're giving you, but we filled the rest of it out so that you can see what we would put if you were to go through this paper and try to see how to fill out the table, how to interpret the figures.
Right, so this is the method, the conclusion, and none of these things actually have to do with showing the activity, which we've already shown. But things like this, these quinones and coumarins inhibit, these are in fact the things, the kinds of drugs that I was telling you about. It uses anti-topoisomerase drugs for particular things. Then there's stuff about the physical properties of the complex, which doesn't say anything about the function. You can't cut them up in little pieces and have them still work. And so on. Okay, so that's an example of just a paper that is a relatively straightforward one. But you can see that there's, of all these figures that are in this paper, the majority of them are actually not going to be things you're going to use for function. So part of the skill in this is going through these papers and figures and figuring out which are the relevant things that you should focus on. Because if something's not relevant, we don't actually spend a whole time on it. At least for this. Now, if you're a researcher who's actually studying topoisomerase 4, you might care about all these other things. Okay? But from the point of view of what do these things do? Yeah, you really don't care about those particular figures. Now, some of the earlier figures, there are things like the sequence. They don't tell us anything about the activity, but they're important for things like, how do I know what gene I'm actually working with? Okay, so one of the key things in doing these papers is you're going to be associating a functional term with a particular gene product, and so you have to know exactly which protein you're working with. Make sure you get it from the right species. It turns out there's going to be times when that's harder than you think it should be because authors can be really sloppy. They assume, oh, everybody works on what I work on. Of course it's a mouse protein. Of course it's a human protein, right? And sometimes they'll mix them up in the same figure and switch on you. You have to be careful about stuff like that.
The one thing I just want to point out is thinking about the way that Dr. Hu walked through that paper. So he kind of knew a little bit of background. He made sure that the big picture was clear, like what is a topoisomerase? That's like the main thing. So he kind of defined terms that you needed to understand to be able to read the paper, and that would have all been stuff you find in the introduction. And then he skipped the materials and methods. Did you notice that? Didn't look at that at all. So he went straight to the results section and started looking at the figures. Now it helps that he'd already seen the paper and he knew what it was talking about, but that's kind of an approach that professionals use when they're reading papers in their field. They often go straight to the data because every figure should contain all the information you need to interpret the figure without reading what the authors wrote in the results section, where they tell you what they want you to think as opposed to you deciding what you think about the data. And so it's fine to read through the results to see what the authors have to say about their data, but it's also something you want to think about, whether you want to try to just look at the figure, look at the data, see what you can get out of it, decide, yes, this bar is different from this bar, or this green's over here and this green is not over here, and then see what the authors say.
Yeah, it's a good point that I skipped over the methods, even though one of the things we're going to have you do is sort of part of the annotation is saying, this gene has this function based on this evidence, and the evidence is tied up in the methods. But what I tend to do is, as I'm going through the figures and I'll see some method that gives some sort of inference, and then I'll go back to that part of the methods in the paper to see, okay, how does this apply to this figure? I'll look at the results and see if it says, okay, as described in this method. Sometimes, in fact, often, what's in the paper is not enough to understand the method that's there. Because often it'll reference some previous paper, which will then reference a previous paper. So sometimes you have to do this little scavenger hunt where you're digging back into older papers to figure out the methods.
So you dig into the methods as needed, and then the discussion part you can read if you need to. It's the only part that I'd say you definitely need to read in the discussion is usually that first paragraph or two where they kind of recap what they did in the paper, and oftentimes the rest of the discussion is talking about the author's opinion about their study and how it relates to how they think it might relate to other things in the field. And so that's really interesting if it's something that you are going to keep reading about, or if you want to know what they think about what is in their paper, but it often isn't something that you should go straight to. You probably want to get the background that you need from the intro and then start looking at the data. But if from the beginning you think you just want to read straight through the paper, sometimes you'll pick up enough terms that you can then go back and kind of do a more directed deep dive into the paper. So don't be surprised if you read this paper, you want to set aside a good amount of time to be able to actually understand what you're reading and look up the things that you need to. Definitely Google terms that you don't know. Wikipedia is a perfectly fine place to get some background to start.